Attention, please.
Why we should experiment on the human genome through the scientific and ethical lenses. Scientists have been able to alter and make changes to DNA since the 1970s, but in recent years they have developed cheaper, faster, and more precise methods to add, remove, or change genes to living organisms. Genome editing is a way of making changes to specific parts of a genome, and researchers are working to develop therapies that use gene editing to treat children and adults for a range of conditions, some including sickle cell, hemophilia, some forms of blindness, and even cancer. These new technologies often raise controversial but essential ethical questions about their unknown possible risks and benefits. Gene editing is a very complex process, but with technologies such as CRISPR-Cas9, the process has become much easier. CRISPR, clustered irregularly interspaced palindromic repeats, is a gene editing technology that allows scientists to be able to make changes to DNA more precisely than ever done before. Segments of DNA occur naturally in bacteria, where they store information to help recognize possible mating viruses. Associated enzymes such as Cas9 can cut viral DNA out of bacterial genes. Scientists are then able to provide a new DNA sequence to replace the cell that was just cut. Although this new technology may seem flawless, CRISPR can sometimes misrecognize a DNA sequence that is extremely similar to the one it's looking for and actually end up cutting in the wrong spot, causing an off-target mutation. Other times it may cut in the right space but cause mistakes or indels where DNA may be inserted or deleted incorrectly. Researchers have already used CRISPR to fix genetic diseases in animals, to combat viruses, and to prepare pig organs for human transplants. Research on editing the genomes of early human embryos, eggs, and sperm has also taken place. Currently, human genome technologies are being explored on a wide variety of single gene disorders and holds many promises for the prevention and treatment of more complex and prevalent diseases such as cancer, heart disease, HIV, and mental illness in the future. All this scientific information leaves the question, is the process of editing the human genome ethical? Currently, most of the changes introduced with genome editing are restricted to somatic cells, which are cells found in the human body other than egg and sperm cells. This means that the changes to the DNA only affect certain tissues and are not passed down from one generation to another. However, changes made to germline cells, which are genes in egg, sperm, and embryo cells, are passed down to future generations. These are 4 categories that discuss the possible ethical issues with genome editing. Safety is a concern due to the possibility of off-target mutations and mosaicism, which is when some cells carry an edit but others do not. Many people are also concerned with the fact that it is impossible to obtain informed consent for germline cell editing because the patients who are affected are the embryos and offsprings of future generations. Another ethics concern is justice and equity, as there is a possibility that genome editing will only be accessible to the wealthy and cause an increase in the already existing disparities in access to healthcare. Lastly, the use of human embryos in research is a very controversial topic. Many bioethical and research groups have stated that a possible solution to this concern is genome editing research on non-viable embryos that are left over from IVF or embryos that are solely created for this research. Experimenting with the human genome is a complicated, controversial topic. However, the possibilities could be very beneficial and life-saving for many. Although gene editing for the enhancements of normal human traits such as intelligence, hair color, or eye color is not ethical, the outcome that CRISPR and other human genome editing technologies to treat diseases and illnesses needs to be taken advantage of.
Thank you. What evidence did you gather that you didn't include, and why did you choose not to include it?
I tried to gather a small amount of information about the cost of genome editing and how it might be a possible ethical concern, but I decided not to use it because I thought it'd be easier to talk about how gene editing may only be accessible to the wealthy, or in general, just to give an idea of it.
Okay, thank you. What are the implications of your findings to the community?
Based on my research and information I gathered, it looks like human genome editing is making many improvements and advancements and could be available in the somewhat near future. This means that the early stages of genome editing or trials are not too far away and could begin happening at places like the Mayo in my community. This would impact others in my community as they could participate using this new technology soon.
Okay, thank you. Another round of applause.